Alzheimer's has been studied for a long time. We actually know a lot about the disease, but to this date, there is no therapy that will stop the progression of the disease. Our research is really able to look at a new pathway that we could use to target Alzheimer's disease with the idea of a faster way to test drugs that would be more likely to be effective in an Alzheimer's patient. We have a new study coming out in the journal Stem Cell Reports where we used stem cells that we made from Alzheimer's disease patients and also patients that didn't have Alzheimer's disease to look at how proteins are trafficked in cells. So the two main proteins that we think cause the problem in Alzheimer's disease are called amyloid beta and tau protein. So we were looking at how these proteins accumulate, and the way they accumulate is they have to get moved around inside of cells in little structures called vesicles. That's very important for how the cell functions. We created these cell models by taking skin cells from subjects that either had Alzheimer's disease or didn't. We used a process called reprogramming, which turns them into a cell called an induced pluripotent stem cell. This type of cell has the potential to become any cell type of the human body. We can make these cells turn into neurons that are very similar to the cells in the patient's brain. So we were able to use gene editing in these stem cells in order to molecularly regulate how much of one of these proteins, amyloid, was generated. So we could induce a mutation in a cell line that was from an individual who did not develop Alzheimer's disease. We could use gene editing and give them a mutation that was similar to a patient who gets Alzheimer's disease very early in life. These cells generate much more of amyloid. We could also use gene editing to completely remove the gene that causes amyloid. So we have a cell line that has a lot of amyloid and a cell line that has no amyloid. Then we were able to look at the other protein, tau, and look at what the effects are of having a lot of amyloid or no amyloid on the second toxic protein. And what we found was that the molecule that stabilizes this important trafficking pathway within neurons worked equally well under both conditions. Our findings suggest that there are pathways in cells that haven't been targeted yet for drug development that we can target that will have a benefit early on in the way these toxic proteins are generated. Our findings also suggest that we can use human neurons to discover these pathways and also use human neurons to test them.